Our next section is going to focus on the three types of muscle tissue. And coming back to the main page for our slide box, let's scroll down to, now it would be there, chapter 4 for muscle. And the first muscle we're going to look at is going to be skeletal. And they have lots of different skeletal muscle sections here. This one looks like an entire muscle group. But yours is going to have, your slide is going to have a longitudinal and a cross-sectional view. So let's go ahead and click this because this is also an H&E stain. And so again, at a low magnification, you can see that we have a cross-sectional view with lots of different muscle sections, meaning like these fascicles that we can see are these collections of cells. It kind of does look like a typical cut of meat in kind of a cross-sectional view. And then we also have our longitudinal section here. So let's start with our longitudinal section first and zoom in. And what we're looking for are these relatively large, long tubular cells that have many nuclei per cell. And also, as we can start to see here, the striations are going to start to pop up. And so this is like a nice, beautiful view. Any of this really is quite nice. You can see this long kind of tubular cell here. You will notice, again, these multiple nuclei per cell, which is formed in development, honestly. So you have lots of myoblasts that basically fuse together to form these really huge, massive cells. And therefore they have like multiple nuclei, which works for the cell. And the striations that we see are these dark and light bands, which represent the sarcomeres that are basically the organization of protein on the inside of our skeletal muscle cells. And so when we're saying that these are striated, we're saying that the protein is arranged in such a way that allows us to see these stripes, right? And so even over here, you can actually see the Z lines. And so you see kind of dark and light bands. And if you find focus with your slide, you really need these really big fat muscle cells, first of all. So, which I think are the newer slides, the newer white tag slides. So if you're zooming in, and play with the diaphragm lever and the fine focus, and you should be able to see the Z lines pop out. And so even down here in between this kind of like light band area, you'll see a dark line, and those are the Z lines at the end of the sarcomere. So from this dark line here, and I know you guys see like a circle, like the yellow circle, so I'm trying to kind of put it right at the center. This dark line in the light band, and then you'll see another dark line here at this light band, and that literally is from one Z line to the next. And so this is the light band, or the I band. I and light kind of go together, think of it that way. And then you also have the A band, which is kind of the dark band. And again, if we go back to our typical sarcomere format, right, we can understand the kind of overlap of our light and dark proteins, or actin and myosin proteins. So the actin is kind of a reminder for all of you anatomy phys people that our actin is the light, thinner protein fibers, and then our myosin is going to be darker and thicker. They try to blow the view up over here, and it's kind of difficult even because it's pretty blurry, and I'm kind of interested to see what they're going to show us. If we click on that, what are they? That's kind of just what we're seeing anyways. These actually do look a little bit nicer. You can actually see lots of the Z lines that are there, lots and lots of nuclei. There is a little bit of connective tissue in between the cells themselves, and this would actually be the endomysium that wraps around the outer surface of each skeletal muscle cell. So we're looking at these in longitudinal form, so we really don't see like the specific mysiums, right, that epi, peri, and endomysium, which we will definitely head over here and look at in a cross-sectional view. So the connective tissue that is in between the cells also have, fibroblasts, right? So that would be kind of like a fibroblast that would be making a lot of that protein fiber support. And if I zoom out a little bit here, I'm just curious to kind of see, ooh, that was too far. What else we can identify? You might find blood vessels running through here. That's absolutely possible. I don't feel that we would have, let me go to the periphery. Yeah, this is just kind of a individual section or individual cut of muscle. I don't think we have an outer surface of an epimysium that is there. But again, I'm just going to kind of click in here, and what else can we really identify? I think overall, the fact that where we are in magnification is something to note. So when you are in even like a 4 power, you will notice if we put skeletal, smooth, and cardiac muscle right next to each other, that these cells, these skeletal muscle cells, are absolutely huge, right? They're very tubular in nature. The striations are so much bigger than what we would see in like a cardiac muscle cell. And also remember, we do have different animal slides, right? May have like human or rabbit or something of that nature, so they might look a little bit different. So again, try to compare your slide to maybe a couple of the other ones in the lab, and just to get a different view of how these striations can pop up into view. What I'm going to do is kind of pause and bring up the idea of the sarcomere from a typical type of anatomy physiology diagram. So this is a diagram that I believe I put somewhere either in your lecture notes or in a handout for lab. And you're looking at a transmission electron micrograph. So this is, obviously much more in detail than what we would be able to see in the lab, but we have this really basic diagram here showing you the sarcomere. And so the light band or the I band is at the end of one sarcomere where another one is starting. And so it basically overlaps two. And so this is that whole light striation that you see on our slide. And then here would be another light striation that you would see. And then here would be the center dark one. Okay, so you can see, especially on that slide that we just looked at, we can see this dark line and this one. We can see the light area, and then we can see a dark. So if I jump back over here and kind of zoom in, let's see, like over here, let's say, you can see again this kind of dark line going between the light and another dark one going between the light, and then you have just kind of a generic dark band that is there. And so this really is the dark band, and then here's the light. And then again, that dark line is the Z line to Z line. So from one Z line to another, that's just one individual sarcomere. And so the dark area is just kind of this entire region. So you don't really see the M line that would be coming down the center. You just kind of see it as a generic kind of dark space. Okay, so just to kind of give you that reference so that you have that there. So let's kind of head over here to the cross-sectional view, and let's zoom out. Let's see if I can get all the way out there. Okay, so this is a nice view of the muscle in cross-section. And you can already start to see that the muscle is, I don't want to say disarticulated, but it's kind of pulled out a little bit. And that allows us a little bit of ease, actually, to see where the breaks are in the actual muscle tissue. So if this was like real tissue, you would not have all this white space. It would be nice and kind of compact together. But the breaks in the tissue do allow us to see how the muscle cells are actually being organized into sections of cells or kind of clusters of cells. And these clusters of cells, as we can see here, like here's a whole cluster of cells, and here's another, and here's another. And there can be smaller and kind of larger clusters. And but regardless, we use the vocabulary of a fascicule to refer to these kind of clusters of cells. So let's just pick any one of these really. And we can start to identify the fascicule. We can identify the cell itself. We might be able to see some of the myofibrils that are there. And then we really start to try to identify some of the connective tissue also that helps to support all of these areas. So the white spaces really are the areas where our connective tissue, our fibrous connective tissue would fill in to help support the muscle cells. So at the outer periphery, so if this was an entire muscle group, at the outer periphery is where you would find the connective tissue that we would call the epimysium. And epi is on the outer surface, my is for muscle, and again, IUM, EUM, it's usually in reference to some sort of a membrane or a kind of outer coating of some sort, right? Usually cellular, like epithelial and connective tissue. And so if we go in with a higher magnification, do we really see connective tissue out here? We really don't. And so I'm wondering if maybe one of the other slides that they're offering, especially that whole muscle group, may have an epimysium that is there. But then you can start to see some of the stringy material, and that really is the connective tissue. The connective tissue that would start to kind of infiltrate and wrap around, let's say, an entire fascicle would be what we reference as the perimysium. So the perimysium wraps around individual muscle fibers. I'm going to go up here a little bit. And even like this kind of big triangular one, it would literally be in this space here. And so some of you might have this issue where you can see muscle in cross section, but you really don't see a lot of connective tissue. So maybe for some reason it just kind of was washed out of the slide. Well, that really doesn't help you. That can be kind of confusing because you're like, where is it? Does it exist? Is it here? And you can see little bits of traces of where that connective tissue was. And if we continue, and maybe that's why we'll pull up the other slide too, but as we continue in here and we kind of go in higher magnification of the cells, like each one of these is representing an individual muscle cell. So this would it's almost at a little bit of an oblique angle, but it would be coming out of the screen towards you, kind of like a giant straw. Okay, so we're kind of looking at the top of the straw. So the connective tissue that wraps around each individual muscle cell, and we also use the word fiber for muscle cell, so muscle cell or muscle fiber, which I think is very confusing, would be that endomysium. So the endomysium connective tissue would wrap around each one of these cells. So let's just go to another area. These are kind of more in the proper orientation. You can see that this is just one cell. You can see all of the nuclei that are also associated with each individual cell. Little bits of connective tissue that are here. Again, here's a little bit more wrapping around. So that's a really nice view of some endomysium. If we could look at, maybe go out a little bit here. You know, we can see kind of the breaks, the larger breaks in the tissue. Like this whole section would be like a fascicle. Okay, and here's like another fascicle. So this would be more kind of filled with fibrous connective tissue. Let's take a look at this guy, because you can see on the inside, you can start to see little dots a little bit, and those all represent the protein fibrils, the actin and myosin that fill in the actual skeletal muscle cell. If it has a little bit of an oblique angle to it, you should actually start to see some striations. So really it's that longitudinal view is where you see striations, and the cross-sectional view is where you can see fibrils from the entry point, not the side view, but the entry point, and also where you can identify lots of the mysiums, is in this cross-sectional view. You would also, in the connective tissue around here, would be able to see, let's jump around a little bit. I just don't think it's going to be here. You would be able to see some of the blood vessels as well that infiltrate muscle tissue. So again, some fibrous, at least you can get an idea of what this looks like. So why don't we do this? Why don't we jump back out? Let's look at this muscle section. And, oh, they do. They have them right over here. Perfect. So this is an entire muscle group. And at the periphery, you can already start to see some of that fibrous nature, and that is going to be your epimysium. So even if we were to kind of click over here, let's see. Oh, you can actually see a little bit of fat there too. So this connective tissue sheathing here is your epimysium. You can start to see it coming inward and kind of separating all of these fascicles here. So as soon as it starts to come into the muscle group, this would be essentially your perimysium, okay? And as it continues down this way as well. You can see a lovely fascicle right here, just a collection of muscle cells. And, if we were to go ahead and click in, you can see that the cells are much closer together at this point. And any connective tissue around here would be essentially, again, your endomysium. You might see blood vessels. That looks like a little capillary to me. Look at that nice little circle here. This looks like a little, oh, look at that. There's a little, that looks like a little venule. That might be a little arterial that is there. You could find fibroblasts. There could be satellite cells that are here too. Here, let's click on capillary and see where it takes us. So, oh, here's a bunch of them. So little capillary, capillary, You know, all of these guys are little capillaries. Sometimes you'll see an endothelial cell, like a simple squamous cell. Sometimes you won't. But know that the membrane goes all the way around. Let's see where they take us for endomysium. That's beautiful. All that little stringy connective tissue there. That would be all endo. Let's see where they take us for peri. So all of this is going to be your perimysium. You can see it cutting up in there. So all of that is peri. And then let's see where they take us for epi. And they're right out to this peripheral border, right? So here is your epimysium. Let's see. Oh, they even kind of point out the two types of fibers, which is interesting. So you can see the different coloration that is here, and they're just showing you the different muscle types, right? We have fast twitch and slow twitch, and we have different sizes of muscle and the muscle cells, right? So they can be smaller or larger in diameter. So there's a lot of other things that we can identify that is not necessarily on your checklist. I don't want to give you too much. But we're kind of at the highest magnification, but what you can see are all of these little dots, right? All these little teeny tiny dots are all of the fibrils, again, in your muscle cells. So this is the border. This is the membrane of your muscle cell. And so the material inside of here, I get this kind of confusion, like, are these the cells? Is this the cell? You know, because it's kind of like all of these tubes inside of tubes inside of tubes, and that can be confusing. But look at the magnification. We are at the 40x, right? We are really in there. You can also compare this back to like a capillary, which is this guy. So you can see that there's the simple squamous nuclei. This is like maybe 1 or 2 simple squamous cells. And that gives you an orientation size, right? That you kind of like can compare the size of the muscle cell in cross section to this guy over here. And plus also, look, these are like fibroblasts, right? This is connective tissue fiber. So that sizing orientation becomes really, really important. You know, whether we're looking at blood or we're looking here at muscle, to be like, okay, this is one individual muscle cell. These are the fibrils. This is the non-living protein, but the whole cell, right, is a living unit. And here's another, and here's another, and another. So hopefully that was helpful in looking at both a longitudinal and the cross-sectional view of your muscle. Other slides that you can go to, so if you have a slide that doesn't have a lot of connective tissue, you can try to head over to, let's say, like maybe a muscle bone connection slide. You might be able to see some muscle at the periphery of like your intervertebral disc, or maybe further outside the trachea, just because of it's kind of a section of the neck. We will run into skeletal muscle in some other areas, but you should have a slide at least that says skeletal muscle and cross section and longitudinal. And then you're also welcome to, you know, look at other slides in the other boxes as well. Just make sure that you put those slides back where they belong. That's all I have for you at this point. And then we will also have some videos for smooth and also cardiac muscle.